Carbohydrates by Allie, Kayla, Kathleen, Jihan, and Michaela. Carbohydrates are required by all life on Earth, so the question of their history is not so much where they began, rather it is what is the history of human knowledge on carbohydrates. From our studies, we know that as early as 6th century BCE, Indian physician Sushruta discovered the link of sweet urine and rice. Up until 1838, sugar and starch were the words used to describe carbohydrates, and in the early 19th centuries, sugars were named by their source, such as grape sugar or sugar cane. Later in the 19th century, a consensus was reached for naming sugars with the ending OSE. Glucose was named in 1838 by John Baptist Andre Dumas. In 1840, Anselm Pam named cellulose. Glycogen was discovered in 1856 by Claude Bernard. In 1884, Emil Fischer began his Nobel Prize-winning work synthesizing glucose and 30 other sugars. In 1919, Otto Meyerhoff showed that glycogen is converted to lactic acid when muscles are working.
So carbohydrates are classified into three groups called monosaccharides, oligosaccharides, and polysaccharides, depending on the size of the chain. And there are also a couple more of carbohydrates called triose, hexose, and pentose, which are 3 carbons, 6 carbons, and 5 carbons respectively. The simplest of the sugars are called trioses, like glyceraldehyde D and L isomers. And the carbonyl, there's also another classification which involves the carbonyl group. So for example, if there's an aldehyde attached to the carbohydrate, it's called an aldose. And if there's a ketose attached to the carbohydrate or monosaccharide, it's called a ketose. Okay, so aldose versus ketose. An aldose is a monosaccharide that has a carbonyl group on the terminal carbon in the chain. And the test for the aldose is called a Selye-Vanoff test. And the aldose will produce a light pink color in the reaction for the positive test. And an example of an aldose is glucose. Uh, ketose is a monosaccharide that contains a carbonyl group on the inner chain, and in the Selye-Vanoff test, a ketose will react rapidly, giving a deep red color. An example of a ketose is fructose.
D and L configuration is the stereochemistry of carbohydrates. To assign this configuration, you first draw a Fisher projection formula with the functional group at the top. Then you count the carbons down the chain starting at the top. And then the second highest carbon is known as the penultimate carbon. And then you look at the hydroxyl group off of that carbon. If the hydroxyl group is on the right, it's the D configuration. If it's on the left, it's the L configuration, as shown in the image. Most biological sugars favor the D configuration, while amino acids tend to favor the L configuration.
Epimers are a type of stereoisomer that differs in the configuration at a particular stereogenic center. If the atoms of interest are on different sides, the compound is deemed alpha, and if they are on the same side, they are deemed beta. Shown in figure 4 are two configurations of the molecule D-glucopyranose. As seen in the left configuration, the hydroxyl group on C1 and the hydroxymethyl group on C5 are on opposite sides of the ring. Thus, C1 is said to be an alpha epimer. In the right configuration, however, these groups are on the same side, making C1 the beta epimer.
Carbohydrates are linked by glycosidic bonds, but in order to understand these, you have to first understand how the ring of a monosaccharide is formed. This is done through the formation of a cyclic hemiacetal, as shown in the figure. A cyclic hemiacetal is only formed when there's a hydroxyl group and an aldehyde in the same molecule. The oxygen of the hydroxyl group attacks the carbon of the aldehyde. The carbon that was originally a part of the aldehyde is now known as the anomeric carbon. Now, getting back to the linkages, they can be defined as alpha or beta bonds. An alpha bond is formed when both carbons involved in the bond have the same stereochemistry, and a beta bond is formed when carbons have different stereochemistry. Cellulose, amylose, starch, and glycogen are all polysaccharides that are made of glucose units, but the units are bonded together in different ways. Cellulose is composed of beta linkages between the anomeric carbon of one glucose and carbon 4 of the next glucose, making it a beta 1,4 bond. Each glucose molecule is rotated 180 degrees, which gives rise to the linear nature of cellulose. Amylose is very similar to cellulose, but it has alpha 1,4 linkages. The glucose molecules are not rotated 180 degrees, so it forms a more bent shape than cellulose. Starch is actually composed of two polysaccharides, one being amylose and the other is amylopectin. Amylopectin is structurally similar to amylose in that it is mainly made of alpha 1,4 bonds, but it also has branching off of carbon 6 in some of the molecules. Glycogen is alpha 1,4 linkages, but it has much more branching off of carbon 6, making it more dense than the other polysaccharides mentioned.
Artificial sweeteners. Despite studies in the 1970s that linked saccharin to bladder cancer in lab rats, the National Cancer Institute says there is not any sound evidence that artificial sweeteners approved for use in the United States cause cancer or any other dangerous health issues. Figure 6 is of saccharin. Cell recognition and blood typing. Cells that are covered in complex carbohydrates are able to carry information for cell-to-cell recognition. This results in adhesion between the carbohydrates and specific cell receptors. For blood typing, oligosaccharides are attached to red blood cells through glycosidic bonds. These oligosaccharides end in a fructose sugar and can have specific enzymes attached to give a different blood type. Figure 7 shows the different enzymes attached to the chain giving certain blood types, such as A, B, AB, and O.
Most of us have heard of low-carb diets that Atkins diet and the ketogenic diet through media like magazines, such as the one in the figure. But just how great are they? Some advantages include weight loss and their usefulness in metabolic syndrome control and high blood pressure control. However, these diets cause a shortage of fiber, which is necessary for colon health, insulin regulation, and proper digestion. Also, low-carb diets may cause gastrointestinal issues, vitamin and mineral deficiency, and bone loss. Diabetes occurs when one's blood glucose levels are too high due to the body's insulin levels. As seen in the figure, insulin assists in the cell uptake of glucose. With type 1 diabetes, the body cannot produce insulin because the immune system attacks cells in the pancreas that produce it. In contrast, with type 2 diabetes, the body cannot produce or use insulin effectively. This condition can cause fatigue, increased thirst, nausea or vomiting, dry mouth, frequent urination, hunger, and blurred vision. Treatments for diabetes include insulin injections and metformin, which stops glucose production. Additionally, by counting carbohydrates, individuals with diabetes can prevent kidney disease, nerve damage, blindness, and blood vessel disease problems that can cause diabetics to have to undergo amputations.
The only structural difference between ribose and deoxyribose is that there is a hydroxyl group on carbon 2 of ribose, whereas deoxyribose only has a hydrogen. You can see this in red in the image to the right. The hydroxyl group on the ribose actually makes it unable to form a stable double-stranded molecule in RNA. It also makes RNA more susceptible to hydrolysis than DNA. It does, however, allow RNA to fold back on itself and form loops, and this becomes important in protein synthesis.
These are some of the real-life applications of carbohydrates that include antibiotics, vaccine production, and there's a new drug-resistant bacteria that is prompting researchers to find other ways to combat bacteria. And the carbohydrates vaccines are also being researched upon. There's also a new approach to test for COVID-19 that is being developed, one that does not use a genetic code of the virus, but instead uses its reliance on the chains of sugar, which, unlike the genetic code that can change, are unchangeable and constant. There's also autobrewery syndrome, which is rare. And this is when a lot of ethanol is made by bacteria that ferments in the gastrointestinal area. The patients that have this report a high carbohydrate and high sugar diet. Thank you so much for listening to the presentation. And these are all the references that have been added to the presentation at the end. And